Okay, this is chapter 24, the urinary system. We've already done the lab, but now we're going to go through the lecture. Hopefully after this we'll start to get caught back up. So the components of the urinary system, they are the kidneys. You have two of those that filter the blood. Some people only have one. You only really need one kidney. You do have to adjust your lifestyle choices, but you can live with one kidney. They remove waste products and convert filtrate into urine. We'll see that blood comes in through the renal artery, it gets filtered into filtrate, and then it becomes urine after a process. The ureters, you have two of those, one from each kidney, that transport urine from the kidneys to the bladder. Your bladder is an expandable muscular sac. We'll see that muscle is called the detrusor muscle. The bladder can store as much as 1 liter of urine. Once it gets to that point, you really have to go to the bathroom. After about 250 mils, that's when you get your first signal that you need to go to the bathroom. And you get one about every 200 to 300 mils of urine more. And so once you get to 1 liter, you really don't have much of a choice after that. You're going to go to the bathroom. You have one urethra that eliminates urine from your body. So as filtrate is converted to urine, like I said, it goes through a couple different processes, and those encompass these. The elimination of metabolic wastes, regulation of ion levels, your sodium, potassium, calcium, regulation of acid and balance through secretion and reabsorption, selective secretion and reabsorption of hydrogen and bicarbonate, regulation of blood pressure, and elimination of biologically active molecules, meaning hormones and drugs. It also forms calcitriol, which helps to increase blood calcium levels, produce and release EPO, which is crucial in regulating red blood cell production, and the potential to engage in gluconeogenesis, creating glucose from non-glucose sources. Has two regions of functional tissue here, so these are structural areas, but they are also functional. These are structural organizations, but these are functional areas, meaning that there's actual processes related to the kidney's function going on here. You have your outer cortex and then your inner medulla. Medulla is in the middle, just remember that always. You have your renal columns, which are extensions of the cortex going down into your medulla, and the cortex, the columns rather, will divide up the renal pyramids. Your lobe is the pyramid and its adjacent renal columns. Your renal sinus, remember that a sinus is just a hole or a space. You have the sinuses in your skull that are spaces to lighten it, to allow for sound and airflow and for mucus flow. Sinuses and the kidneys are going to be storing and collecting urine. They are your minor calyces, major calyces, and your renal pelvis. The minor calyces merge to form the major calyx, and renal pelvis is formed from merged malar calyces. You can just see that depiction. There you have your outer cortex, this inner medulla. Right, once you get into this area, this is your renal pelvis, where all of these major calyces are kind of coming together. Maybe it's definitely easier to kind of see it on this one. This yellow stuff is urine, by the way. Yeah, I think you'll have lab images. Some of the lab models that we have also have these little things right here, and that's urine. So innervation of the kidney. Each one is innervated by both the sympathetic and parasympathetic divisions. The sympathetic nerves are going to be activating the blood vessels of the kidney and the juxtaglomerular apparatus. This will decrease urine production. So as you have sympathetic innervation, the more sympathetic innervation you have, you will decrease your urine production and cause vasoconstriction. The parasympathetic nerves from your vagus nerve, we don't really know what the effects are. There is innervation, but we don't know. And sometimes this happens as you get more and more into specifics into anatomy. This is one of those that it seems it's a very kind of large subject, so you would think that we would know. But as you get more and more specific into anatomy topics, you kind of realize, oh, there's some things that we just don't know yet. You'll have textbooks where it's just a question mark in a box when it comes to labeling something or identifying a process because somebody's still working on their PhD for it. All right, so functional, you have your nephrons, collecting tubules, and collecting ducts with their associated structures. We'll see them. A nephron is a microscopic functional filtration unit of the kidney. This is the essential functional unit of the kidney. This is where the filtration and secretion transformation into urine happens. It consists of the renal corpuscle and the renal tubule. The renal corpuscle has two structures to it, the glomerulus and the glomerular capsule. There are several different names for these. You'll see a lot of times Bowman's capsule, the nephron corpuscle I've seen, so there's other ones. Renal corpuscle, that's what we call it. Inside of it, you have the glomerulus, which are the capillaries. Remember, a glomerulus is just your renal capillaries, your renal capillary beds. And the glomerular capsule, that's what actually surrounds it. When you have these two things together, that makes the renal corpuscle. So that the glomerulus, tangle of capillaries, Blood enters through the afferent and exits through the efferent, arrives afferent, exits efferent, E for exit. The capsule, again, it's called also Bowman's capsule. It has the internal permeable visceral layer that actually is overlying the glomerular capillaries. So this is the permeable one, which will allow blood to be filtered out as filtrate. And the external impermeable parietal layer, which will not allow things to be filtered, things not to be filtered. Between those two layers is the capsular space. So similar to how your lungs have the parietal layer on the thoracic cavity and the visceral layer on the actual lungs themselves, your parietal layer is on the capsule, which allows things to not be filtered out of that. So blood is filtered through the glomerular capillaries into the capsular space, and then prevented from leaving there by this impermeable parietal layer. The capsular space receives that filtrate, and it becomes modified to form urine. Not within the capsular space, it has to go through the tubule system for that, but that's what collects it. There are two poles in a capsule. You have your vascular pole, which is the afferent and efferent arteriole, so vascular vessels. So on the side of the capsule that has the vessels, that's your vascular pole, and then on the side that has the renal tubule that leads to the tubular system, that's your tubular pole. So the tubule consists of three continuous sections: your proximal convoluted tubule, your nephron loop that has a descending and an ascending limb, and then your distal convoluted tubule. The proximal convoluted tubule is the first region of the renal tubule, so as you exit out of the capsular space, filtrate is going directly into the proximal convoluted tubule. There are microvilli that increase the surface area and reabsorption capacity. So we'll get more in depth into all of these regions, but just go ahead and note that the proximal convoluted tubule is the main site of reabsorption in the tubular system. The nephron loop, again, has a descending and ascending limb. Descending immediately from the proximal returns to the renal cortex and ends at the distal convoluted tubule. It has both thick and thin segments that we'll see alter their permeability to water and sodium to help with concentrating urine. Note that the descending limb is first. So as it comes out of the proximal convoluted tubule, it goes down the descending and then up the ascending into the distal convoluted tubule. It's not ascending then descending, descending then ascending. After the loop, you have your distal convoluted tubule. It originates in the renal cortex, into the ascending limb, extends to the collecting tubule, and it's lined by simple cuboidal epithelium without microvilli, so not as much reabsorption happening, very little. Here is just a basic structure of that. So you can see you have your vessel here. It's a division of the renal artery that comes in through the kidney. It branches off into this afferent arteriole that goes into the capsule and becomes this capillaries. So this is an arteriole, so that's the smallest version of an artery. It's going to divide once more and become a capillary. Here's your capillary bed, your glomerulus, and then those will merge to form your efferent arteriole. Notice that the afferent is larger than the efferent. That's going to help maintain that pressure gradient to help blood be filtered to provide hydrostatic pressure. Once that forms, you have your proximal convoluted tubule, and then you have your nephron loop, your descending and then ascending limb, and then it comes back up into the distal convoluted tubule. Notice that the distal convoluted tubule is still close to the capsule. It's not distal because it's farther away from it. It's just distal because it's further along in the tubular system. From there, you have your main collecting duct that you have these several collecting tubules draining into. So all of these collecting tubules are going to be coming from the distal convoluted tubule. This is one of my, I think, favorite images from this chapter, just because it goes ahead and puts everything in there. You should know all of these regions. You should know what type of reabsorption, secretion, filtration is happening, what is being secreted, what hormones are being activated in these areas, what cells are doing the secretion and activation. I have, this is mine. So yeah, it looks just like this. So here is the distal, here's the proximal. So these are all the things that get reabsorbed and secreted. These are waste products over here. This talks about sodium and water reabsorption. I've kind of labeled the portions of the loop that are impermeable, permeable to water. And then again, down here, all the regulated reabsorption. And then in the back, I talk about the specific cells of the kidney, as well as the hormones that are regulating secretion and reabsorption. So you can do all of that based off of this image. Okay. Two types of nephrons you have. You have your cortical and your juxtamedullary. So based on two factors, the relative position of the renal corpuscle in the cortex and the length of the nephron loop. So remember, the cortex is the outer portion. The nephron loop is going to go down into the medulla for whether they're cortical or juxtamedullary, but the juxtamedullary ones we'll see go deeper. The cortical nephrons are short loop, barely penetrating the medulla. These are 85% of your nephrons. So most of your nephrons are going to be these cortical short loop. Juxtamedullary have a long nephron loop going deep into the medulla to help establish salt concentration gradients in the interstitial space, in that space outside of the tubular system, to allow for regulation of urine concentration. So these long juxtamedullary ones, even though they're only 15% of the nephrons, they really play a major role in actually being able to concentrate urine and water balance. So here you have a cortical nephron. You can see that the capsule is higher up in the cortex. Its nephron loop barely goes into the medulla, versus a juxtamedullary one. Again, the capsule is juxtamedullary. It's right next to the medulla, and it's going to go deeper into the medulla itself. You can also see that it has these vasorecta capillaries going around its loop, where this one did not. These capillaries are going to assist in that in the urine concentration process. Okay, collecting tubules and then collecting ducts. So nephrons drain into a collecting tubule from that distal convoluted tubule. Remember, you have multiple collecting tubules emptying into that larger collecting duct. Numerous collecting ducts empty into the papillary duct. These are, you have special epithelial cells in your distal convoluted tubule, your collecting tubule, and your collecting duct. Both principal cells and intercalated cells, and there are two types of intercalated cells. The principal cells are responsive to hormones aldosterone and antidiuretic hormone, ADH. And intercalated cells are both specialized epithelial cells to regulate pH and blood pH. We'll see that they do that with hydrogen and bicarbonate regulation. Your juxtaglomerular apparatus, let's talk about this. It's there to help regulate blood filtrate formation and systemic blood pressure. So it helps to regulate the rate at which blood or urine is being formed, as well as systemic blood pressure throughout your body. The components are, you have granular cells. These are modified smooth muscle cells of the afferent arteriole, located near the entrance to the renal corpuscle. So they're the afferent arteriole, the one that's going into the renal corpuscle. And they contract when they're stimulated by stretch or sympathetic stimulation. They synthesize, store, and release renin. So what these are going to do, if they contract, that means you're going to be decreasing your urine production. You're going to decrease the amount of filtration that's formed. So as they contract, they're going to be slowing down the rate of blood. So not only are they decreasing the amount of blood that's entering the glomerular capillaries, they're also starting to decrease the pressure, the hydrostatic pressure within the glomerular capillary itself. Because if they slow down the flow of blood coming in, that will decrease the hydrostatic pressure, which decreases the filtration, the rate of filtration into the capsular space. And again, that's sympathetic stimulation. It's going to cause decreased urine production. As well as the granular cells, they synthesize, store, and release renin, which we'll see also increases blood pressure. Because as you contract the afferent arteriole, it will decrease the blood flow and blood pressure within the glomerular capillaries, but it will help to increase your systemic blood pressure, which sympathetic stimulation does. The other component, your macula densa cells. These are modified epithelial cells in the wall of the distal convoluted tubule. I talked about in the lab lecture, macula densa distal, D and D. So just that's how I remember those. These are located on the tubule side, next to the afferent arteriole. So still kind of close in there with the granular cells. They detect changes in sodium chloride concentration, the sodium chloride concentration in the fluid of the lumen of the distal convoluted tubule. They signal granular cells to release renin through paracrine stimulation. You should remember paracrine stimulation is a type of endocrine stimulation where you're signaling your neighboring cell. These will signal to release renin to help increase blood pressure. If your sodium chloride levels drop, if your salt levels start to drop, that means that you're not going to be reabsorbing as much water, which is going to lead to a decrease in blood volume, which leads to a decrease in blood pressure. So by signaling, if they detect decreased sodium chloride concentrations in the urine, or at this point it's still filtrate, the DCT, if they start to detect decreased amounts of sodium chloride, that means that there is going to be increased amounts of water and decreased blood volume. So that's their way of monitoring blood pressure. And filtrate formation, that's part of filtrate formation, is making sure that there's an appropriate appropriate concentration of sodium chloride. The third part is the extraglomerular mesangial cells, mesangial cells, however you say them, just outside the glomerulus, in the gap between the afferent and efferent arterioles. They communicate with the other cells, but their function isn't well understood, but they're there. So here you can see just the graphic representation of it. You have this larger afferent arteriole feeding the glomerular capillaries, and this efferent one that drains them, that drains this capillary bed. Within here you have your juxtaglomerular apparatus, the macula densa cells on the distal convoluted tubule, and then the granular cells on the afferent arteriole. Okay, this is just a blood supply flow to the kidney. It comes in through the renal artery, and then it starts to divide. It becomes the segmental, interlobar, arcuate, interlobular, so interlobar, interlobular. And then from the interlobular artery, that's when it starts to enter your capillary bed. It becomes your afferent arteriole, glomerulus for the glomerular capillary, and then it becomes your efferent arteriole. And here's where it starts to switch. Depends on if it's a cortical or a juxtamedullary nephron. If it is a cortical nephron, it is only going to have paratubular capillaries. If it is a juxtamedullary nephron, meaning it has that long nephron loop, it's going to have both. It's going to have both paratubular and vasorecta capillaries. The paratubular just wrap around the tubular system, the proximal and distal convoluted tubules. The vasorecta will actually wrap around the nephron loop, like we said, for urine concentration. So if you have a cortical nephron, the efferent arteriole, as it exits out of the glomerulus, will just go around the distal and proximal convoluted tubule. And this is just for, so that way there is blood flow for secretion and reabsorption, just for regulated secretion and reabsorption. Because in the glomerulus, we'll see filtration just happens based on pressures and protein concentrations, and some selective filtration based on the membrane permeability. But once it actually starts getting to regulating hormone levels, ion levels, and things like that, blood pH and things like that, there has to be some type of connection between the bloodstream and the tubular system, and that is these vasorecta capillaries, paratubular capillaries that are in all types of nephrons. When you have your juxtamedullary nephron, you have to be able to concentrate urine, and so it also has the paratubular nephrons. Those two systems, capillary systems essentially, will then come to the interlobular vein, arcuate vein, interlobar, renal vein, drains back into the abdominal aorta. Your arterioles and capillaries. The afferent enters the renal corpuscle, forming the glomerulus. Some blood plasma becomes filtered there. Efferent exits. It branches into the paratubular vasorecta, like we said. The paratubular intertwined around the PCT and DCT, primarily in the cortex. Vasorepta associated with the nephron loop, primarily in the medulla of the kidney. All blood moves through two capillary beds in the kidneys. It gets filtered to the glomerular capillaries once, and then it passes through the paratubular capillaries or the vasorecta for gas exchange, nutrients, or waste. Finally, they all become drained into a network of veins. So blood that goes to the kidneys, all the blood in the body, essentially then, will become filtered twice, is what it's basically saying. Not filtered twice, but pass through capillary beds twice. Okay, so your filtrate. So as blood is filtered, as blood flows through the glomerulus, water and solutes are filtered from your blood plasma, and it moves across, those filtered components will move across the wall of the glomerular capillaries into that capsular space, forming the filtrate. We'll see that this has unique components to it. It's not just, whatever we'll see. Okay, your tubular fluid, new name for filtrate once it enters the proximal convoluted tubule. So it's just based on location at this point. There's no difference in composition. It's going to be tubular fluid as it flows through the proximal convoluted tubule, the loop, the distal, the collecting tubules, the collecting ducts. Once it empties into the larger collecting ducts, that's when it is called urine. There's really no more secretion or reabsorption at that point. From there, urine enters the papillary duct within the renal papilla. It flows within the renal sinus of the kidney, which remember is your minor and major calyces, into your renal pelvis. The pelvis connects to the ureter connects to the bladder, and the bladder will hold it and excrete it from the body through the urethra. So there is your whole urine flow. Here is a nice little picture of that. You should know all these steps, locations, and what the fluid is called at each point. Okay, let's get into the process of forming urine. Urine is formed through three interrelated processes: filtration, reabsorption, and secretion. We start with glomerular filtration. This happens in the glomerular capillaries. It separates some water and dissolved solutes from the blood plasma. Notice it's not all of water gets filtered out, it's some water. Goes into the capsular space of the renal corpuscle. It is due to the pressure differences across the filtration membrane. So if you did not like the pressure, the net filtration pressure equations from the chapter 20, then you probably won't like it here either. You have your water and solutes in the capsular space. That separate fluid is filtrate. After that, you have tubular reabsorption, movement of components within the tubular fluid by diffusion, osmosis, or active transport, moving from the lumen of tubules and the collecting ducts across the walls of the tubule system. Return to blood within peritubular capillaries and vasa rectus. Remember, those capillary beds are going to be wrapping around the tubular system, and then they'll join back into the renal vein eventually, carrying with them any solutes or any molecules that need to be reabsorbed. All vital solutes and most water is reabsorbed in your urine, or I'm sorry, during this process. So even though some of it will get filtered out, a lot of it gets filtered out. Most of the vital ones, we'll see a lot of the sodium, glucose, and water is reabsorbed. Excess solutes, waste products, and some water will remain in your tubular fluid. And then finally, you have tubular secretion. This is the movement of solutes, usually by active transport, into the lumen fluid. It moves out of the blood within the peritubular and the vasorectal capillaries into the tubule system. Materials move selectively into the tubules to be excreted. Remember, if something is secreted, that means that it's going to be moved from the blood into, in this case, the lumen of the urine, the tubular lumen. If it is excreted, that means that it is leaving the body and is not going to be able to be reabsorbed. If something is secreted, there is always a potential that it could be reabsorbed, but if it is excreted, that means that it is leaving the body. So at this point, these are going to be excreted. This means that it's secreted, but excreted. Secreted to be excreted. That's a fine distinction, but it's an important one. Here is a nice little representation for that. Filtration, reabsorption, and secretion. Again, you can see even down into here, you do have some reabsorption happening kind of later into the process, but it's very selective at this point. Really, most of the reabsorption is going to be happening at the proximal convoluted tubule, and then it gets modified as it travels through the system. Okay, so let's look at the filtration membrane within the glomerulus. It is porous, thin, and a negatively charged structure. So it's negatively charged. We know that opposites, or that same in this case will oppose, so negative will oppose negative. What is one of the primary negative components in your bloodstream? Your proteins. Great, that's correct. So this will help to resist, not only because it's small and thin, or sorry, not only because it's small, the negative charge of the protein of the membrane will resist protein filtration, which is good. You don't want to have protein in the urine. That's a bad thing. It's formed by the glomerulus and the visceral layer of the glomerular capsule. You have the endothelium of the glomerulus is the first part. It is fenestrated, allowing plasma and dissolved substances to pass. So it's fenestrated, which means it has holes, but it's not sinusoidal, meaning that it has those large gaps where formed elements could pass. It's going to restrict the passage of larger structures like your erythrocytes and some larger proteins. It has a basement membrane, which is the glycoprotein and proteoglycan molecules, restricting the passage of large plasma proteins. And this visceral layer, which is the outermost layer wrapping around the glomerular capillaries, has specialized cells called podocytes. These have long processes called pedicels that help support the capillary wall but don't completely enclose it, and provide filtration slits that help to restrict the passage of most small proteins. So the way that I describe this in the lab thing is that if you think about, like, if my finger were like a capillary and my hand is a podocyte, so this is the cell body and my fingers are the pedicels that will wrap around the capillary. And in between my fingers, you can see these little filtration slits. So if each finger is a pedicel, each space between my finger is a filtration slit. If they constrict, you have less filtration. If they dilate, and the way that it works with these, and it's a little bit different, is that with the podocytes, if they relax, that means that they kind of swell and their filtration slits will become smaller and you'll have less filtration. Whereas if they contract, they draw back and they expose their filtration slits so that more fluid can be filtered out. So it's just a slight difference there. They have to contract. That draws their pedicels in and increases the size of the filtration slits, allowing for more filtration. Still restricting most proteins. The mesangial cells are phagocytic, contractile, and have signaling proteins, or properties rather. They're between the glomerular capillary loops, helping with the process of filtration. So here you can see your main podocytes, its little pedicels wrapping around, there's filtration slits. You have your fenestrated endothelium. This one's a little bit more detailed, I believe. You have in here this filtration membrane. The endothelium right there, the basement membrane with its filtration slits. Okay. Filtrate includes water, glucose, amino acids, ions, urea, some hormones, vitamins B and C, ketones, and very small amounts of protein. You should know what is normal composition for filtrate as well as for urine. Okay, so this is not necessarily what is going to be excreted from our body, but you should still know what the normal composition, what normal filtrate is, and what should be typically blocked from filtration. What will happen to some of these small proteins, like the ones that get stuck in here, the mesangial cells will come in and eat those if stuff gets trapped in the filtration membrane. So you produce about 180 liters of filtrate a day. We already said it's filtered plasma with certain solutes and minimal amounts of protein caught within the capsular space and funneled into the proximal convoluted tubule. What's not filtered remains in blood and exits the renal corpuscle through the efferent arteriole if it isn't secreted out at a later point. Some of the filtered membrane, like I said, trapped, and the phagocytic mesangial cells will eat it. So here is what is freely filtered: your small solutes, water, glucose, amino acids, ions, those type of things. These will pass easily through your filtration membrane. So you should expect those to have no problem. Remember, ions, things like sodium, potassium, calcium, magnesium, zinc, whatever. More ions. Those are the main ones, right? Chloride. I think that's the only one I didn't name that's simply main. So not filtered at all. Formed elements and large proteins. If you start seeing blood in your urine, you know that's a bad thing automatically. If you have large amounts of proteins or large proteins in there, that's not going to be great. Too much, like certain hormones should not, some will, but if you have large amounts of hormones in your urine, that's a bad sign too. Mainly because they can't pass through the filtration membrane because of its size and its charge. Limited filtration, proteins of intermediate size, so smaller ones, usually blocked from filtration due to the size or negative charge. Right. So what are the pressures associated with glomerular filtration? What's actually driving this process? You have your glomerular hydrostatic blood pressure, your HPG. This is the pressure within the glomerulus, within the capillary bed, within the capillaries, that's going to push water and some of the solutes out of the glomerulus. So as blood pumps from your heart, it goes all the way down to these glomerular capillaries, and as that pressure as blood goes into the afferent arteriole, the pressure from that is going to be pushing the filtrate. Higher than blood pressure of other systemic capillaries because it's required for filtration to occur. That larger diameter of the afferent arteriole versus the smaller diameter of the efferent arteriole. We saw that with the lymph nodes. Pressures opposing the glomerular hydrostatic pressure. You have the blood colloid osmotic pressure and the capsular hydrostatic pressure. So the osmotic pressures exerted by dissolved solutes in the blood, so there's plasma proteins within that fluid that are not being filtered out. They're staying in the blood. And as you have filtration occurring, it's going to be pushing water out, but these larger proteins are going to be staying in the blood. That will increase their colloid osmotic pressure, their relative colloid osmotic pressure, which will then try to resist that filtration. Even though you have fluid being pushed out, these proteins are going to be resisting that draw of water, or you'd be drawing water back into the glomerulus. Simultaneously, as the water, as filtrate is being formed from the glomerulus into the capsular space, that's going to increase the pressure within that capsule. So it's going to increase your hydrostatic pressure of your capsule. That will impede the movement of additional fluid. So during net filtration pressure, if the pressures promoting filtration are greater than pressures opposing, then you will have a positive NFP, as you see here. That's really the big thing with it, is if it's a positive NFP, you have filtration. If it's negative NFP, you have reabsorption. Here's just a representation for that. So if your hydrostatic pressure of the glomerulus pushing filtrate out, colloid osmotic pressures, and the hydrostatic pressure of the capillaries drawing fluid back in, Ultimately, you have a net 10 out. Okay, variables influenced by your net filtration pressure. So, by the way, if I'm swallowing it, that is weird. I'm using the AirPod as a microphone, so please let me know if the sound is weird with this, that I just need to not use this or do something different. Okay, your glomerular filtration rate. This is the rate at which the volume of filtrate is formed. You might connect this it is directly related to your NFP. As NFP increases, GFR increases, and vice versa. But we do distinguish them between two different things. So the glomerular filtration rate, volume per unit of time. Increased net filtration pressure increases GFR. It increases the solutes and water remaining in your tubular fluid. Increases substances in urine, and decreases filtrate reabsorption. So what happens when you increase your GFR and increase your net filtration pressure is that when you increase your NFP, you're not just increasing how much filtrate is formed. You're not just influencing the beginning of the system or the beginning of the process. You're influencing it all the way down the chain. So if you have an increase in GFR, an increase in that rate of filtrate being formed, it's going to increase the rate that fluid is moving through the tubular system. You know, like when you have to use the bathroom, but you also feel kind of thirsty, so you drink a little bit of water, and you drink some water, and even though you know that water has to move through your entire body and be absorbed before it can get to your kidneys, somehow drinking that water feels like it's moving urine faster through your system. So we can increase our GFR, and it will push fluid. It does not, by the way. Drinking more water is just maybe it increases pressure on your bladder, but it's not actually going to be speeding up the process of urine formation. But increasing your NFP will increase your GFR. That will increase the flow rate of fluid through the tubular system. If your flow rate is faster, just like we saw in capillary beds, you aren't going to have as good of exchange. So not as good of exchange of substances. So you're going to have increased solutes and water remaining in the tubular fluid because it won't be able to be reabsorbed. Increased substances in the urine in general, just because there won't be as much, because you're having this increased pressure that can put more strain on the filtration membrane, which could make it filter out larger proteins or more varied molecules. You won't also have that time for reabsorption to happen. So you end up just with a larger amount of urine and potentially with more stuff in it. For this reasons, GFR is tightly regulated. It helps the kidney control urine production based on physiologic conditions, your hydration status, sodium chloride levels, pH, those kind of things. GFR is influenced by changing the luminal diameter of the afferent arteriole, so constricting or dilating that arteriole, and altering the surface area of the filtration membrane, so contracting or relaxing those mesangial cells to increase or decrease the filtration sleds. These things can be done by the kidney itself through intrinsic controls, or there are extrinsic controls that can regulate this. The renal auto-regulation, those are your intrinsic controls, right? So auto-regulation, renal, so it's the kidney's intrinsic control system. It's the ability to maintain constant blood pressure and GFR throughout the kidney. Because that's the goal, right? Is that we should have, if everything is working normally, we should have a constant rate of GFR and blood pressure within our kidneys, so that way they're not having these large spikes or dips in pressure. Maintains the spikes of changes of systemic arterial pressure, so even as you're running and exercising and increasing your blood pressure, or laying on the couch for 4 days and just not getting your heart rate above 60, then your GFR and your kidneys should still stay the same. It functions by two mechanisms. We have a myogenic response, which we have already seen, and the tubulo-glomerular feedback mechanism, which we've talked about a little bit. Your myogenic response, if you remember, is contraction or relaxation of the smooth muscle of the afferent arteriole in response to stretch. Decreased blood pressure equals less stretch of the smooth muscle in the arteriole. So as your blood pressure decreases, it's going to be pushing less blood into that afferent arteriole, which will decrease the stretch on that vessel. This causes the muscle cells to relax and the vessels to dilate to allow more blood into your glomerulus, compensating for that lower system pressure, letting your GFR maintain its normal rate, right? So you have a lower BP, which slows down the systemic blood pressure, the systemic blood flow rate, Once that gets into your kidney, it's going to be even slower. We know that blood slows as it divides into capillaries, and so your afferent arteriole will dilate to allow increased blood flow. That's a resistance thing. Remember, as the resistance decreases, flow increases. So here we're having a decrease in resistance and an increase in blood flow that will help us to maintain GFR. With increased blood pressure, more stretch of the smooth muscle in the arteriole happens. That causes them to contract. The vessels constrict, less blood flow into the glomerulus, compensating for that greater systemic pressure. GFR still remains normal. So this is just the opposite. If you're increasing blood pressure, if you have a higher blood pressure, that's going to stretch those arterioles more. They'll contract and resist that expansion, that stretch, and resist that increased flow. This is just a nice picture representation. The tubular glomerular feedback mechanism, mouthful, is the backup to the myogenic mechanism response to increased blood pressure. So if glomerular blood pressure is increased, the amount of sodium chloride in the tubular fluid is also increased. So remember, we said that if you increase your GFR, if you increase your NFP, you're increasing your GFR. If your GFR increases, you're increasing the amount of stuff that is filtered, as well as the amount that is decreased. You're increasing the amount that's filtered and decreasing the amount that's reabsorbed. So this will increase relatively your sodium chloride in the tubular fluid. The macula-densa cells will detect that in the juxtaglomerular apparatus and results in further vasoconstriction of the afferent arteriole to decrease filtration, to decrease GFR. As you increase your sodium chloride, that means that you're going to be increasing your water loss. So your vessels will vasoconstrict to compensate for that. Yeah. Okay, limitations to maintaining GFR. Renal autoregulation, it maintains normal glomerular pressure when the blood pressure is within a certain range. So from 80 to 180, your renal autoregulation system can maintain normal GFR, as long as your blood pressure is maintained in between those levels. Decreasing it below 80, the arterioles become at maximum dilation. The decrease in the glomerular blood pressure and the GFR, if extremely low, you can have cessation of waste elimination in urine. So once your blood pressure starts to dip below a certain amount for a prolonged period of time, you can have a decrease in urine function, or a decrease in kidney function. Increasing blood pressure above 180, the arterioles will be at maximum constriction. The increase in glomerular blood pressure and GFR, meaning that your urine formation will be increasing. This also, though, means that you're going to be increasing your loss of solutes and fluid and decreasing reabsorption once you get to this point. For a prolonged period of time. If you're exercising, that's normal. If it's for a short period of time, that's fine. But if it's for prolonged extended times, it's not good. So there is kind of this normal mean arterial pressure, kind of an average pressure range for when urine production is maintained by renal autoregulation and will be optimal. Okay, the extrinsic controls, neural and hormonal. These involve physiological processes to change GFR, in contrast to renal autoregulation, which attempts to maintain GFR. Okay, so those are differences. Decreasing GFR through sympathetic stimulation during exercise or emergency results in decrease in GFR through vasoconstriction of afferent and efferent arterioles. The granular cells of the JG apparatus release renin, which results in angiotensin II production and contraction of the mesangial cells, which we'll see how that impacts filtration. And contraction of the there it is. Contraction of the mesangial cells decreases surface area of glomerulus, decreasing GFR. I think I might have said that backwards earlier. Yeah, I think I did. Okay, decreasing surface area of the glomerulus, decreasing GFR. So as mesangial cells contract, they decrease the GFR. The body therefore conserves water under stressful conditions. Okay. Increasing GFR through ANP. It's a peptide hormone released from cardiac muscle cells in response to stretch of the heart. We've seen this one in chapter 17, chapter 19. So as ANP, as blood, you have increased venous return to the right atria. It's going to increase the stretch. Those cells will release ANP. That will increase your GFR through relaxation of the afferent arterioles, so trying to release out more, or increase the GFR, right, and increase the urine formation. It inhibits the release of renin, causing relaxation of the mesangial cells. Relaxation of the cells increases filtration membrane surface area, increasing GFR. Net increase in GFR with increased urine volume decreases blood volume and overall blood pressure. So you should understand how these hormones work. Again, I refer you to a nice little chart. So this is the sympathetic stimulation to decrease GFR. I'm just reading over that real quick. Yeah, you should know pretty much all these little steps. Increasing GFR through A and P, you have vasodilation of the afferent arteriole and relaxation of mesangial cells. And here's the RAS system, the renin-angiotensin-aldosterone system. Your liver is releasing angiotensinogen. Kidney releases renin. They meet in the blood, and that becomes angiotensin 1. That circulates through the pulmonary circulation, as where it encounters ACE from the liver, a little bit from the kidney, I guess, and that converts angiotensin 1 to angiotensin 2. Angiotensin 2 will then go on and have, you should know its effectors and their effector responses. Overall, it's going to be increasing blood pressure, but you should know specifically how it's going to be affecting each one. Constricting glomerular efferent arteriole, increasing sodium-hydrogen exchanger activity, that will all influence blood volume. Posterior pituitary to secrete ADH, vascular smooth muscle hypertension, so vasoconstriction, stimulating the thirst response in the hypothalamus to increase blood volume, and secreting aldosterone, which also increases sodium absorption. Okay, so yeah, you should know all these things that influence GFR, and how GFR plays a role in urine inflammation, and how increasing or decreasing GFR changes the composition of urine as well. Okay, so transport processes, paracellular and transcellular transport. Paracellular movement of substances between epithelial cells. Transcellular is moving them across the epithelial cells, so going through them. They must cross the luminal membrane in contact with the fluid, the basolateral membrane, and the basement membrane, and the order depends on whether it's being reabsorbed or secreted. So it's either to go from the lumen to the basement, or lumen to the cell if it's being reabsorbed, or cell to the lumen if it's being secreted. You have transport proteins embedded within the luminal and basolateral membranes controlling the movement of various substances for facilitated transport or active transport, depending on what it may be. And the peritubular capillaries, the low hydrostatic pressure and a high oncotic pressure, that facilitates the reabsorption of substances through bulk flow. So the peritubular capillaries are fed directly from that efferent arteriole, so you've just lost all the water, some of the ions, but you still have all those plasma proteins. So that's going to want to draw things back in. You have the negative, it's a more negative charge in your blood, so it's going to be drawing in some of those more positively charged ions, and with those ions, blood flow or water flow will happen as well. Most reabsorption of the PCT aided by microvilli, increasing the general surface area. So here you can see the tubular fluid moving down. Paracellular transport moving between the cells. Transcellular transport, again, going through the epithelial cell and then through the basement membrane. Just trying to see if there's anything. Yeah, there's the transport proteins, channels, carriers, pumps that are facilitating that transport. Okay, transport maximum and renal threshold. This is a fairly important concept. So transport max is the maximum rate of substances that can be reabsorbed or secreted across the tubule epithelium per a certain time. It depends on the number of transport proteins in the membrane. So they can become exhausted, they become overwhelmed, and at capacity essentially if you have a high enough concentration of a certain substance in your blood, if it's a hormone or a drug or some kind of toxin. There's only, at a certain point, there's only so many proteins that can allow for that exchange facilitation to happen. If there's no more than 375 milligrams per minute of being, or if there is no more than that, that needs to be filtered. Glucose in the tubule is all reabsorbed. If you have more than that, if you have an excess of glucose, then it's excreted in the urine. So for some diabetics, that's one of the things that they'll always look for is glucose in the urine, because it means that it's not, there's excess in the blood because it's not being taken up by the cells. And because there's excess in the blood, it can be excreted out in the urine. So basically that's your transport maximum for glucose is 375 milligrams per minute. If you have more than this amount in your urine, in your blood, then it will be excreted. So that's the transport maximum. That's the amount, and then we have the renal threshold. The renal threshold is the max plasma concentration of a substance that can be transported in the blood without appearing in the urine. If the TM is exceeded, the substance is excreted in the urine. So what it's saying is that there is only so much that you can have transported across the lumen and be. Let me go back to this one first. I don't know why these are here. I want them to be here. So the renal threshold says that this is for kind of like drug regulation, right? So depending on how much of a certain drug you should take, or let me get my thoughts straight before I start talking too much. So when you're being dosed for a certain drug from a pharmacist or a doctor, they will look at how quickly that substance is cleared out of your urine and how much they can give you that will be able to stay in your blood. So for certain things, there is a certain concentration that will be allowed to circulate within your bloodstream without being excreted out in your urine. You can have a certain base level of a drug for a period of time, stay circulating in your bloodstream, and because it's a low level enough, you're not reaching its transport maximum. Because there's a low enough level, kind of base level of that drug or of that hormone, we're just keep saying drug, in your bloodstream, it is able to be all reabsorbed. There's enough transport proteins that can reabsorb that back into your bloodstream, and so it can stay in your body longer. If for some reason a doctor were to overprescribe you a medicine, if they gave you too much of it, you would exceed your renal threshold, which means that you've gone beyond that maximum plasma concentration, and now it's going to start being filtered in your blood because you've overcome the transport maximum. So the renal threshold refers to the plasma concentration, whereas the transport maximum refers to how much is exchanged within the kidneys. Okay, the transport maximum, that's all about the transport proteins and the ability to be able to move a substance across the membrane. If you have too much of a substance, it's kind of like a backup at the airport. If you have too much of a substance, some people are going to miss their flight and they'll have to go on to the next one, and they'll just kind of keep traveling down the urine, so the tubular system. Renal threshold is just saying that at a certain point, sort of like vitamin C, that's a great example. Vitamin C is a water-soluble vitamin. You will urinate it out if you take too much of it. There's only so much vitamin C that you can take in a day, and at a certain point, and this is true with all vitamins, at a certain point, if your body isn't absorbing those vitamins that you're taking, it's going to excrete them because you've reached your renal threshold. A lot of times people, same thing, there's tons of examples with this. Now I got all the examples. People that want to go work out, they'll drink protein shakes, but then not go to the gym, and they won't build any muscle. And all of a sudden, they just have all of this extra protein that they're consuming. Their body's going to eliminate it somehow. It's going to convert it usually down to glucose fat and then store it, or it may eliminate it somehow in the body. If you have too much of a substance circulating in your bloodstream and your body isn't actively using it and taking it up into the cells, it's going to pass through the kidneys and be filtered. And if there's enough of it, if there's an excess of it, it will pass its renal threshold and potentially pass its transport maximum and be excreted out. Okay, going on. So substances that are reabsorbed completely. Your nutrient reabsorption, normally all reabsorbed. So each nutrient has its own specific transport protein, similar to how when we saw partial pressures of gases, each nutrient is going to have its own specific transport protein as well. So for glucose transport, it's transported into a tubular cell by sodium glucose symporter proteins. If you don't remember, a symporter is one that will basically import or will move two things in the same direction. It will kind of use one to help move the other. Use one of the concentration gradients to move the other one. Moved by uniporters out of the tubule system. And then glucose is returned to blood in the peritubular capillaries, 100% reabsorbed in a healthy individual. So from the lumen of the tubule system to the across the basolateral, from the lumen into the bloodstream, or from the lumen to the cell. I'll get there eventually, y'all. From the lumen to the cell, you have a symporter, and then moving out of the cell through a uniporter into the peritubular capillary, 100% reabsorbed in a normal person. So here is your lumen, here is your epithelial cell, here is your bloodstream. You have a symporter, a glucose-sodium symporter, both pumping them in. Again, you can see you have this low sodium concentration in the cell, a high one out here. So glucose basically just kind of hitchhikes in there with sodium. And now you have this high glucose concentration, which is going to pump it through a uniporter, so just pumping glucose out of this epithelial cell into the bloodstream. Okay, Proteins, most not freely filtered due to their size and their negative charge that fights against the membrane. Some small and medium-sized ones may appear in the filtrate. Small amounts of large proteins, very little, transported from the tubular fluid in the proximal convoluted tubule back into the blood. So this happens in the PCT, whereas this one was happening, I believe, mostly in the DCT, or in the PCT as well. Protein moves across the luminal membrane by pinocytosis and receptor-mediated endocytosis, digested by lysosomes or peptidases, so it gets broken down into amino acids, and that is those move by facilitated fusion back into the blood. I thought it was going to give us a thing, but it did not. So pinocytosis, that's basically cell eating. They kind of make a vesicle and bring it in, similar to kind of receptor-mediated endocytosis. There's a receptor on the cell that helps to transport the protein in. Once the protein gets into the epithelial cell, it's going to be digested by different types of enzymes to help break it down into its constituent parts, which can be easier moved into the bloodstream and transported. For sodium, 98 to 100% of sodium is reabsorbed from your tubular fluid. It's reabsorbed along the entire nephron tubule. Most of it is in the PCT. The concentration is relatively low inside the tubule cell, higher in the tubule membrane, tubule lumen, sorry, and the interstitial fluid that's around it. Sodium moves down its gradient across the luminal membrane into the tubule cell. Sodium-potassium pumps embedded in the basolateral membrane keep sodium relatively low in the tubule cell, but do require substantial energy. Sodium-potassium pumps do for short. Reabsorption is regulated by hormones, aldosterone and ANP towards the end of the tubule. But because our dietary intake of sodium varies significantly, these processes and these the actions are going to be different for each person. It just depends on your diet. For some people, if you have a lot of sodium, then you might reach your renal threshold and your transport max and excrete sodium. So those hormones, aldosterone is a steroid hormone from the adrenal cortex, stimulates protein synthesis of sodium channels and sodium-potassium pumps, bedded in the plasma membranes of principal cells, increasing sodium reabsorption, and then water follows by osmosis. That's that obligate water movement. As sodium moves, so does water. ANP inhibits the reabsorption of sodium in the proximal convoluted and collecting tubules, inhibits the release of aldosterone. You have ending up with more sodium and water excreted and an increase in your GFR. So these two opposing each other. Here's just a nice little overview. 65% of it reabsorbed in the PCT, 25% in the loop, and then facilitated or regulated in the DCT, CT, and CD. That is just a nice little picture of it. Trying to see if there's anything I need to comment on, but I think it is pretty self-explanatory. Oh yeah, there's, remember the sodium-potassium pump? It's pumping sodium out to keep the concentration low in the cell, so more keeps getting drawn in. Once it gets into the fluid, then now the concentration is high in here, and it can just diffuse into the bloodstream. Yeah, and this is just in the distal convoluted tubule. In the latter part of it, you can see your principal cell with the sodium-potassium pumps embedded in the membrane, aldosterone increasing those channels and pumps, while ANP would inhibit them. All right, water reabsorption. It's reabsorbed by paracellular transport between cells, transcellular transport through water transporter proteins, and aquaporins, which are specific aqua channels. 180 liters filtered daily, all but 1.5 liters is reabsorbed. So you do lose some urine or some water in your urine, and ideally your urine will have the color, that whole color should be kind of clear to almost just a pale yellow. Once your urine starts getting darker than a pale yellow and you can smell it, that means you're probably dehydrated and you probably need to drink some more water. Because even though our body will reabsorb most of it, it is going to be filtering out some of it, and there should be enough water in there to allow for that filtration. Because if you have decreased water, you'll have a decreased blood volume, which will decrease your blood pressure and decrease your GFR, right? Or it could inhibit your GFR. Tubule permeability varies along its length. For water, that is 65% in the PCT. Your aquaporins, constant number, maintain a constant number. Water flow follows sodium. In the nephron loop, 10% of the filtered water becomes reabsorbed. Later in the tubule system, water reabsorption is by aldosterone and ADH. Aldosterone increasing sodium potassium pumps and sodium channels, which will draw more water in, and ADH also draws more water in. This is going to bind to principal cells, increasing the migration of vesicles containing aquaporins to the membrane, adding channels to increase water absorption. So basically, they just add more water channels where the principal cells are. The concentration gradient within the interstitial fluid means water reabsorption is regulated by ADH near the end of the tubule, independent of sodium reabsorption. And then you have your tubular reabsorption. So when it follows sodium, that is obligate. When it's independent of sodium, then it's facultative water reabsorption. ADH will continue to increase water reabsorption from filtrate into the blood. It results in smaller volume of more concentrated urine. Elevated levels during dehydration, your urine becomes noticeably darker. With decrease, urine is less concentrated. With decrease of ADH, that is, urine becomes less concentrated. Urine range is 1200 milliosmoles to 50 milliosmoles. There's just that water transport. So obligatory following sodium, and then facultative down here. So here is the facultative. You can see you can have the paracellular transport or transcellular transport through aquaporins. Potassium. So yeah, some of these are where we just basically get into how each of these ions move. That's why I said that for mine. Again, you see I have all of the little ions there and there and there. So potassium is both reabsorbed and secreted, 6 to 80% reabsorbed in your tubular fluid. It is dependent on the movement of sodium as well. Sodium is, all those sodium potassium pumps and sodium channels and things. As sodium is reabsorbed across the luminal membrane, water follows. That increases the concentration of remaining solutes in the tubular fluid. So as the tubular fluid is losing water, the relative concentration of the solutes remaining in the tubule system are going to increase. That creates a gradient between the tubular fluid and the interstitial fluid. Potassium moves down that gradient by paracellular route, allows for passive reabsorption of other solutes as well. So other ones will follow potassium too. 10 to 20% reabsorbed in the thick segments of the nephron loop. Net secretion or reabsorption in the collecting tubule. Intercalated cells reabsorb potassium continuously. So potassium is crucial for maintaining resting membrane potential and cellular and neural cellular activity. So the intercalated cells are continuously reabsorbing potassium. Principal cells secrete potassium at varying rates based on aldosterone level. That stimulates principal cells to secrete it. More powerful stimulant for aldosterone is the elevated potassium levels. So while aldosterone will kind of follow with blood volume and sodium concentrations, elevated potassium levels will activate it even more to secrete potassium from principal cells. So again, you can see how it's regulated. Type A intercalated cells reabsorbing continuously, principal cells varying their secretion with aldosterone levels. Okay, calcium and phosphate balance. 60% of your calcium goes into your filtrate. The remainder is bound to protein and prevented from filtration. 90 to 95% of phosphate is filtered as blood passes through glomerular capillaries. This is important because calcium phosphate is your bones. So when you have both of these together, you can make bones. Parathyroid hormone regulates the excretion of calcium and potassium by inhibiting potassium reabsorption in the proximal convoluted tubule. It's going to stimulate calcium reabsorption in the distal convoluted and inhibiting phosphate reabsorption and stimulating calcium reabsorption. Less phosphate available to form calcium phosphate, calcium deposition in bone is decreased, and calcium blood levels increase. So what it's saying is that it's doing is that your body uses calcium phosphate to make bones. So when you have decreased blood calcium levels, your body will release PTH, which is supposed to increase your blood calcium. Normally when we talk about it, we talk about how it will break down your bones. It will increase osteoblast activity to break down your bones, which will release calcium phosphate into the bloodstream. That calcium phosphate, if it is not immediately taken up, if the calcium isn't taken up, then it will be filtered in the blood. What happens is if you have too high of blood calcium levels, if your blood calcium levels are too low, rather, your parathyroid hormone not only breaks down your bones, it will cause the excretion of phosphate. If you don't have enough phosphate, then it doesn't matter how much calcium you have, you can't make bones. So you kind of have this system where PTH is breaking down your bones to release calcium phosphate into the bloodstream, and then it is also increasing the secretion of phosphate so that calcium is just left freely floating and can be used for other body processes apart from building bones. Does that make sense? It's not so basically it's not only breaking down bones, it's making sure that they can't be reformed. And that calcium can be used to do something else. So yeah, that's the thing. PTH regulation of calcium ions results in increased phosphate loss from urine. Okay, bicarbonate, hydrogen ions, and pH. Bicarb ions move freely across the filtration membrane. It's filtered. Bicarbonate is not reabsorbed. Blood is too acidic, so it will not come back. 80 to 90% reclaimed from tubular fluid. Remaining 10 to 20% taken up from the thick segment of ascending limb. It's this weird thing where filtered bicarb is considered that it is replaced, not reabsorbed. So this whole like filtered bicarbonate, not reabsorbed thing, it's true. It does not get reabsorbed, but basically what happens is it gets broken down and then reconverted and turned right back into bicarb. So the pH of urine and blood regulated in the collecting tubules. If your blood is acidic, meaning it starts to drop below that 7.35, which for us, anything below 7.35 is acidic, anything above 7.45 is basic. If it's acidic blood, then synthesized bicarb is reabsorbed into the bloodstream. Again, synthesized bicarb. Hydrogen is excreted within the filtrate by type A intercalated cells. This will increase blood pH and decrease urine pH. So you're taking acidic blood and making acidic urine and alkaline blood. Notice hydrogen ions are excreted by type A cells. If your blood is alkaline, then type B cells are active. These are going to secrete bicarb and reabsorb hydrogen. By secreting bicarb, they're going to lower your blood pH and increase your urine pH. So 80-90% of bicarb reclaimed in the proximal convoluted tubule, 10 to 20% in the nephron loop, and then is regulated bicarb hydrogen reabsorption. Just want to make sure if I need to read this. Oh yeah, this is that process of reconverting bicarb essentially. So within filtrate, bicarb combines with hydrogen to form carbonic acid. So you can see, right down in here. Bicarb is forming with hydrogen to make carbonic acid. That splits into carbon dioxide and water. That is what technically gets reabsorbed, is the carbon dioxide and the water. Once it gets back into the cell, it's going to form the carbonic acid again. Carbonic acid is going to split. You have a hydrogen that gets pumped back into the lumen, and then you have this bicarbonate that gets transported into the bloodstream. So technically this bicarbonate and this bicarbonate are not the same. Okay, it's how it works. And these are type A cells. So this one, yeah, you can see it's absorbing carbon dioxide, making carbonic acid, hydrogen, and bicarbonate. Yeah. Okay, elimination of nitrogenous waste and waste products. So nitrogenous waste are metabolic wastes containing nitrogen, ammonia mainly, urea. Your main nitrogenous waste products. Sorry, I've been working with fish a lot, so ammonia is on my mind and like the nitrogen cycle. So for humans, you have urea, uric acid, and creatine. Your urea is a molecule produced from protein breakdown. It is both reabsorbed and secreted. We'll see that 50% is excreted in the urine, but then 50% is reabsorbed to help maintain water concentration or establishing concentration in the interstitial fluid for water balance. Uric acid is produced from the nucleic acid breakdown in livers, both reabsorbed and secreted. Creatine is produced from creatine metabolism in muscles and is only secreted. Creatinine. I don't know why I was going to call it creatine. That's a supplement, I think. Creatinine. Okay, eliminating drugs and bioactive substances. Most secretion is occurring in your proximal convoluted tubule, so that's where most of our reabsorption occurs as well. So it's a busy place. Certain drugs, penicillin, sulfonamides, aspirin, other metabolic waste, urobilin, hormone metabolites, as well as some hormones, HCG and epinephrine. HCG, that's what they pregnancy test will test for. Okay, so it's pretty straightforward. These things are all going to be eliminated to a greater or lesser degree. Okay, now let's talk about establishing the concentration gradient. So this is present in the interstitial fluid surrounding the nephron. So remember, you have your nephron, which is either a cortical or juxtamedullary one, but surrounding that is going to be interstitial fluid. This concentration gradient is established by various solutes, sodium, chloride. It progressively increases in concentration from the cortex down into the medulla. So as you go deeper into the kidney, you're increasing that concentration gradient. This exerts an osmotic pull to move water into the interstitial fluid when your ADH is present, right? So if you don't have ADH being present, your body is mostly going to be holding on to and reabsorbing that water, but ADH is going to allow, holding on to the water in the tubular lumen and excreting it. ADH will help us to reabsorb that water. Okay, so the nephron loop provides a positive feedback mechanism called the countercurrent multiplier that involves the nephron loop and helps to establish the concentration gradient. Primarily, your juxtamedullary nephrons are involved. Your descending limb is permeable to water and impermeable to salts. So as water moves from the tubular fluid, water will move from the tubular fluid to the interstitial fluid because you have, it's permeable to water, right? Or it's permeable to water on the descending limb. Salt will stay on the inside. So you have this kind of higher concentration salter fluid on the inside of the tubular lumen than you do in the interstitial fluid. So as it goes down the descending limb, water will move out, but salt won't be able to leave the tubular lumen. Salts retained in the fluid become concentrated. The ascending limb is impermeable to water and salts get pumped out. So it's the opposite. More concentrated the salts, the more is pumped out. So if you have a higher than normal salt intake for most people, and during the descending limb you're pumping out a lot of water, you're going to have a higher concentration of salt in your ascending limb that will draw more water in. You're going to have more salts in your fluid since water can't leave at this point. Salts can only be pumped out. So your tubular fluid will be pumping salts out into the interstitial fluid to try and re-establish that balance. Getting a salt gradient in the interstitial fluid. That's the whole point. In the vasa recta, the blood travels in the opposite direction to tubular fluid of the adjacent nephron loop. So basically you have tubular fluid flowing one way and then blood flowing the other way, and they're exchanging that salt and water. So this is the countercurrent exchange system. It helps to maintain the concentration gradient. Water diffuses out of the vasa recta capillary by osmosis. Salt in the interstitial fluid enters the vasa recta by diffusion. It increases the concentrations of salt in your vasa recta. Next, the vasa recta runs along the descending limb of the nephron, where the gradients are reversed with salt diffusing out and water in. So we have to think about this, that the tubule system, the nephron loop, is running descending to ascending this way, whereas the vasa recta is running ascending to descending. Well, yeah, it looks weird for us, but it's ascending to descending limb for the vasa recta. It's flowing in that direction. So here you can see your tubular fluid movement versus your movement of blood. It's going this way. So as the tubular fluid gets down to here, this is all permeable to water. So you're losing water. That's all going to be kind of drawn into the vasa recta here, into the interstitial fluid. As you're doing that, this is becoming more and more concentrated with salt. Once you go up the ascending limb, you're permeable to salt, which then gets drawn into the vasa recta. As the salt draws into here and it comes back up to this, what is this side permeable to? It's permeable to water. So water is going to be drawing into here. Here, this one's probably better to look at. So being able to understand the permeability of the nephron loop, the permeability and the flow of the nephron loop versus the flow of the vasa recta shows you how water can be, how this can be used to maintain water and salt concentrations. And really, this is one of those systems where you could kind of think about it. The big picture, and I forgot to say this at the beginning of the chapter, but whenever you start to get into the minutiae of this stuff, try to go back to big picture. The purpose of the urinary system, the purpose of our kidneys is to excrete waste and try to maintain as much solutes, nutrients, and water as it can. So that's all that this is trying to do. This is just a way to concentrate urine more fully, to try and draw even more water, even more nutrients, essential nutrients, out of what will eventually become urine. Right now it's still tubular fluid. It's trying to draw as much nutrients out of that as it can and return those to blood circulation. That's all that this is trying to do. It just does it. We call it the countercurrent gradient, the countercurrent mechanism, multiplier, whatever it's called. And yeah, multiplier and exchange, right? So multiplier by concentrating the fluid, exchange by exchanging water and salts. So all of these are doing are just concentrating urine. Let that, if you need to, just step back and take it there for now. Okay, so what are other ways we can establish that gradient? Urea recycling. This helps the concentrating process and the interstitial fluid. For your recycled urea, about half the solutes of the interstitial fluid gradient. Urea is removed from tubular fluid in the collecting duct by uniporters. It diffuses back into tubular fluid in the thin segment of the ascending limb, so it gets pumped out in the collecting duct. It gets reabsorbed into that tubular fluid on the ascending limb of the nephron loop. No, never mind. And remains within the tubular fluid until it reaches the collecting duct again. Urea is cycled between the collecting tubule and the nephron loop. So it gets pumped out of the collecting tubule and goes back into the nephron loop. Once it's in there, it can help with this concentration gradient. All right, summary of reabsorption and secretion. After filtration, the majority or most other substances are reabsorbed or secreted. It happens in your nephron loop, your vasa recta, and your recycling. They're responsible for establishing the concentration gradient of the interstitial fluid to help concentrate your urine. You have the regulation of specific substances during reabsorption secretion, right? Controlled mainly by principal cells and intercalated cells. Those are going to be in your collecting tubule. And the urine is composed of water dissolved. Ultimately, you have urine composed of water, dissolved substances, waste products, drained into your renal sinus of the kidney and excreted by the urinary tract. GFR is the rate that rate filtrate is formed per unit of time, can be measured with an inulin injection. So now we're jumping gear. So that was essentially urine formation. Now we can talk about measuring glomerular filtration rate and detecting kidney function. So GFR can be measured with an inulin injection because it's freely filtered and not reabsorbed nor secreted. So think about this. It's freely filtered, meaning that it is not a protein that's going to be inhibited by the charge. It's not inhibited by its size. As it goes into the glomerulus, pressure will filter it all out. So it is freely filtered. It is not reabsorbed in the proximal convoluted tubule, so we don't have to worry about any of it getting drawn back in, nor is it secreted from the blood. So whatever is filtered in the glomerulus, that's all that's going to be excreted. So if it gets filtered in the glomerulus, it is excreted. There is no more secreted from the capillaries that surround the vessels, and that's important because it allows you to directly measure your glomerular filtration rate independent of any kind of adjustments that may happen later in the tubular system. Urine is collected and measured for volume and concentration. Plasma concentration of inulin measured at given time intervals. So GFR equals UV over P. U is the concentration of inulin in your urine. V is the volume of urine produced per minute, and P is the concentration of inulin in the plasma, in your blood plasma. So normal GFR for inulin is 125 milliliters per minute. Less than this indicates decrease in kidney function. So that's the number that you should remember. Normal GFR is 125 mils per minute. That's how it should be filtered out. This is basically saying that for every minute that your bloodstream is running, it can filter 125 milliliters of this process. Each of your glomeruli can. Okay, renal plasma clearance test. It's another means of assessing kidney function. It measures the volume of plasma cleared of a substance in a given time. The volume of plasma, blood plasma, the fluid portion, cleared of a substance, so how much of a drug or a hormone or whatever it may be of some molecule is removed from the plasma, is filtered out of the plasma over a given time. If the substance isn't reabsorbed or filtered, the clearance equals GFR. If the substance is reabsorbed, the clearance is lower than GFR. Okay. I think this is, don't quote me on this, but I'm pretty sure that's supposed to say secreted instead of not filtered, but I think it's supposed to say secreted. We'll get to this though. But essentially, if it's not reabsorbed, its clearance is equal. If it is reabsorbed, it's lower than GFR. So if a substance is reabsorbed, its clearance is lower than your GFR. We'll get to it. If substance is filtered and secreted, its clearance is higher than your GFR. So what we can say here is that if substance is reabsorbed, that means that it's been filtered out of the glomerulus, and now it's going to be drawn back in. So that means that its clearance is lower than the GFR, so that means that it can, it's able to be drawn back. I have to finish reading all this. We have to get through all these, because until I have the examples and it doesn't work. Okay, if substance is filtered and secreted, your clearance is higher than GFR. For drug clearance, it affects appropriate drug levels. Creatinine clearance, clearance is only slightly higher than GFR, can be used to approximate your GFR rate because it's very close to it. Let me see if, here we go. Okay, so if your substance is filtered and secreted, then your clearance becomes higher than your GFR. So if it has to be filtered, that means it's going to be filtered out of the glomerulus. And then if it's secreted, if it's also secreted, that means that from the vasa recta, from the bloodstream, lower down or further along in the tubule system, more of it is going to be released out. So its clearance has to be higher than your GFR. This means that that rate of 125 mils per minute, if you have a substance that's both filtered and secreted, that means that its GFR is going to be higher than 125 mils per minute. Rather, its clearance is going to be higher than that because that rate of 125 mils per minute, that's kind of the maximum GFR, right? That's kind of our optimal maximal GFR. If your drug, let's say its clearance is 170 mils per minute, that means that if flow rate is running, if your drug clears at 170 mils per minute, but flow rate is at 125 mils per minute, that means that you have this much, that difference of that molecule, of that drug, whatever it be, that is not getting filtered at the glomerulus. So there's this much that then has to be secreted out from the vasa recta into the tubule system. Do you see how that works? Again, this is why it's so frustrating to not have a student right in front of me. Okay, let me go back to this one. So if it's reabsorbed, its clearance is lower than your GFR. So this is talking about your plasma clearance. This is saying that, okay, if let's say GFR is 125 mils per minute, if your molecule has a GFR of, let's say, 70 mils per minute, this means that you have this much that is actually going to be able to be reabsorbed. So there's that much that can then be drawn back in because you have this much that can be held in the plasma without being filtered out. So if your GFR, if you get a GFR for a hormone and it says that it's 70 mils per minute, you should automatically know, okay, that's going to be filtered and reabsorbed. If you have a GFR that is going to be 170 mils per minute, then you should know, okay, that's going to be filtered and secreted. And that's where I want you to work with this. Your GFR, that rate of 125 mils per minute, just says, okay, if a substance is at this rate or lower, it's going to be filtered, but it may also have to be reabsorbed. If it's higher than that, it's going to also be secreted, potentially. Okay. So drug clearance, again, that clearance of drugs being able to not just be in the plasma, but then how much is filtered out. If their GFR is higher or lower, that will affect your dosing rate. Because if it's, and again, this is why it's important. So for drug clearance, if your drug clearance is lower than your GFR, it's going to be reabsorbed. If you have a drug that's constantly having a small amount reabsorbed back into your bloodstream, you don't want to then, and you don't want to give a too high of a dose because you might have too much that gets reabsorbed on top of taking new medicine that could increase it to toxic levels. All right, let's look at the characteristics of urine. Urine is a product of filtered and processed blood plasma. It is sterile unless contaminated with microbes in the kidney or the urinary tract, so any kind of bacteria that might kind of live in there. Characteristics, composition, volume, pH, specific gravity, color, turbidity, and smell. We'll look at all of those. Those are all very important characteristics when diagnosing. Composition should be 95% water and 5% solutes. Remember, we said that so much of the water is reabsorbed, but look, most of your urine is still water. The solutes include salts, nitrogenous wastes, some hormones, drugs, and ketone bodies. 24.4 has abnormal constituents. You should know those. Volume, average is 1 to 2 liters per day. Variations due to fluid intake, blood pressure, temperature, diuretics, diabetes, and other fluid excretions. Sweating, respiration, diarrhea. Minimum of half a liter to eliminate waste from the bodies. So you have to produce at least half a liter of urine to eliminate the normal waste products that your body accumulates. pH is normally between 4.5 and 8, so it varies greatly. More acidic with larger amounts of protein or wheat in the diet, less acidic with diets high in fruits and vegetables, influenced by metabolism and infection. Because of these two things, typically Americans are going to have a more acidic urine. It's not better or worse. I mean, if your urine can be 4.5 to 8.0, like that's a huge range. But just typically, with the way our diets are, more Americans are going to have eat more protein, more wheat, so we have a more acidic urine. Specific gravity is the density of a substance compared to water. Urine is slightly higher than water because of its solutes. Its color ranges from almost clear to dark yellow, depending on the concentration of urobilin. Increased volume of urine, you have a lighter color. So increased volume of urine means that you have an increased amount of water, which is going to decrease the amount of relative solutes. That'll also decrease the specific gravity. Smell is urinoid, normal smell of fresh urine. It may develop ammonia smell if allowed to stand, as urine just sits. I'm sure you have been in a bathroom where someone did not flush immediately. Ammonia is a volatile compound that the urea will convert into ammonia and become that awful smell. I have 2 cats, so I'm very familiar with it. Fruity smell in diabetes, and I believe also in ketoacidosis. Okay, moving to the urinary tract. Your tract are your ureters, your bladder, and your urethra. The ureters conduct urine from the kidney to the urinary bladder, originating from the pelvis as it exits the hilum of the kidney. The wall is composed of 3 tunics, those 3 layers, just like we saw in blood vessels. Not the same, but close. Your mucosa, your muscularis, and your adventitia. Mucosa is transitional epithelium. It's distensible and impermeable to urine. So no urine in or out at this point in the ureter. This is just a transport tube. It folds to fill lumen when no urine is present. So similar to the esophagus, when there's nothing in there, it will collapse in on itself, and then urine will cause it to open back up. It has an inner longitudinal and an outer circular layer of smooth muscles. We'll see that in the digestive system as well. It helps to contract and move urine through the bladder, through the ureter rather, and then into the bladder. The adventitia is the external layer. It's collagen and elastic fibers, just to help anchor it to the supporting structures and give it some type of form and shape. There is your lumen. You have your mucosa, your muscularis, and then your adventitia. Once it goes from the ureters, it enters into the bladder, which is that expandable muscular container, acts as a reservoir for urine. The trigone is a posterior-inferior triangular area of the bladder wall, formed by imaginary lines connecting the ureter openings and the urethra. So you have, let's say you have your 2 ureters coming down here. Here's your bladder. Let's say here's your bladder. The ureters come into here. On the inside of the bladder, where the ureters enter, and then where the urethra begins, that little triangle is called your trigone. And that's essentially where urine sits. Infections are very common in this area, as well as bladder cancer, because urine tends to sit and collect in that area. 4 tunics in the bladder wall: mucosa, submucosa, muscularis, and an adventitia. The mucosa is the innermost, accommodates shape with distension. The mucosal folds allow for that. You have your submucosa with vessels, arteries, nerves, things like that. The muscularis layer has 3 layers of smooth muscle, but they are all kind of intertwined and wrapped around or layered on top of each other, very crisscrossed. So it's collectively just termed your detrusor muscle. It is involuntary, as well as an involuntary internal urethral sphincter. These are formed by smooth muscles encircling the urethral opening, and then an adventitia on the outside. So here's your bladder with your ureters, your trigone in this area. You have your internal urethral sphincter, and then your urethra. So ureters, 2 ureters, 1 urethra. That's just the histology of it. I don't get too big into histology stuff. Okay, from your bladder, then you go into your urethra. This is an epithelial-lined fibromuscular tube exiting the urinary bladder through the urethral opening to the exterior of the body. There are 2 sphincters restricting the release of urine until bladder pressure is high enough. These are the internal and external urethral sphincters. The internal sphincter is involuntary, and it is superior. So it is internal, meaning it is right there. Yeah, right there. Surrounding the neck of the bladder, composed of smooth muscle, and controlled by autonomic nervous system. The external urethral sphincter is inferior to the internal urethral sphincter, controlled by skeletal muscle fibers of the pelvic diaphragm. It is voluntary, controlled by the somatic nervous system, and we learned control of it through toilet training. So usually around 2 to 3 years old. I don't have a child, so that's just kind of what I assume. I hope by 3 that you aren't still in diapers, but we all have our own time. But typically, it is around 2 to 3 years old. You start to learn toilet training and control of those somatic nerves, the pudendal nerve. Did I want to say anything else about these? Not yet, I don't think, because we do the whole micturition reflex in a minute. Okay, you have your females and males both have urethras. In a female, it is a single function, just to transport urine. That's it. In the male, it is a passage for both urine and semen, so it has both urinary and reproductive functions, as well as 3 segments: the prostatic, membranous, and spongy urethra. Really, you should just be able to identify them. There's some little notes about them. The prostatic is the most dilatable. The membranous is the shortest and least. The important part is that it ends with the external urethral orifice. So the external urethral orifice, for both males and females, that's what we can kind of control to close and open, releasing urine in and out. We can't control the internal involuntary one, but the external one we can. There's the male. You can always tell it's a male by a prostate. If you see something sitting underneath the bladder, it's a male. If there's not, it's a female. Okay, so micturition. Micturition is the expulsion of urine from the bladder, associated with 2 reflexes. You have both a storage reflex to store urine and a micturition reflex to release it. These are regulated by sympathetic, parasympathetic divisions, respectively. So innervation of the urinary bladder and urethral sphincters. The urinary bladder has sympathetic, parasympathetic, and somatic innervation. So sympathetic and parasympathetic from the autonomic nervous system, somatic from the somatic nervous system. Sympathetic axons, they're extending from T11 to L2. I'm not so concerned that you remember them specifically, just again that concept that as you go lower down the spinal cord, you're going to be innervating lower structures. The sympathetic axons inhibit contraction of the detrusor muscle and micturition. So this is the storage reflex. If you are inhibiting contraction of the detrusor muscle, you are allowing it to relax. You are allowing it to expand. You're inhibiting micturition. You're inhibiting that urinary, that need to urinate, and so you're drawing all of that back in. Parasympathetic division goes from the micturition center in your pons, so this is brainstem activity. It causes contraction of the detrusor muscle, relaxation of the internal urethral sphincter, and stimulates that micturition reflex in our body. So contracting the detrusor will squeeze it and compress and increase the pressure inside, which will increase the pressure against that internal urethral sphincter. That's going to relax, so urine can move out through the urethra. Then you have the pudendal nerve of the somatic nervous system innervating that external urethral sphincter. It contracts to prevent urination. So as we kind of actively hold urine in and resist going to the bathroom, we're activating our pudendal nerve of our somatic nervous system. So the storage reflex is continuous sympathetic stimulation. Causes relaxation of the detrusor to accommodate urine, stimulates contraction of the internal urethral sphincter so that urine is retained in the bladder. And the external urethral sphincter is continuously stimulated by the pudendal nerve to remain contracted. So when we're younger, before we learn toilet training, we don't have the ability to continuously stimulate our pudendal nerve. We learn it at such a young age that we don't even realize that we're doing it. We don't realize that as our body is, I guess we'll see in this a couple of slides, that, as our bladder fills, we get these micturition reflexes to, you need to go to urinate, you need to go to the bathroom. But we don't realize that, I'm doing this actively. That's why we ask children, well, are you sure you don't need to go? Do you want to go try before you go into a car ride? Because sometimes as they go and just try and they just allow themselves to relax, I do need to urinate, because they don't realize it. We have a better gauge of being able to feel those reflexes and kind of become aware, yes, I am doing this now, or I do need to go urinate. We become more sensitive to those signals. But as we're, just in our normal day, we don't really realize that we're continuously signaling this pudendal nerve, because it's a somatic nerve, it is not an autonomic nerve. We're continuously signaling our pudendal nerve to remain contracted. The sympathetic stimulation, though, it's causing constant relaxation of the detrusor muscle to allow for that accommodation of urine and contraction of the internal urethral sphincter, so urine isn't flowing through the urethra. Okay, during the micturition reflex, the volume of the urine in the bladder goes between 200 and 300 mils. The bladder is just stretched enough and baroreceptors in the wall will be activated. You get a visceral sensory neurons signaled by baroreceptors stimulating the micturition center in your pons. The micturition center alters nerve signals down the spinal cord through pelvic splanchnic nerves. That's always a fun word to say, splanchnic. And then you ultimately get parasympathetic stimulation, which causes the detrusor muscles to contract. Remember, the detrusor muscle and the internal sphincter are both smooth muscle, so you need that autonomic stimulation from parasympathetic division to be able to cause those, 1, to contract, and then 2, to relax. There is just that reflex. Again, the stretch of the bladder will send nerve signals up the spine from the pons, the micturition center, back down the spinal cord and the spine nerves that will innervate the bladder. You can also have conscious control of urination initiated from the cerebral cortex through the pudendal nerve, causing relaxation of the external sphincter, facilitated by a voluntary contraction of the abdominal and expiratory muscles. We'll see this is, we did respiratory system. So this is a Valsalva maneuver, that ability to be able to close the epiglottis over the larynx and increase and bear down and increase abdominal pressure. That pushes pressure on the bladder, which facilitates the movement out, facilitates emptying. After emptying, the detrusor muscle relaxes to accommodate urine again. The neurons of the micturition reflex are inactivated, and the neurons of the storage reflex become activated. If urination isn't activated at the first reflex, relaxation of the detrusor muscle due to the stress relaxation response. So basically that's just saying that, okay, I'm going to get stretched, and then I can, when smooth muscle is immediately first stretched, it'll want to contract and release and contract a little bit to resist that stretch, but then it can accommodate and relax. So, after the first 200 to 300 mils, your bladder will send that micturition signal. It'll want to empty, but if you ignore it, that smooth muscle will start to stretch and expand, and it'll accommodate more urine. But it gets activated again after another 200 to 300 mils. Usually urination occurs involuntary between 500 and 600 mils. I know it said that you can hold up to 1,000 mils, but there's a point at which you're really going to have to go. Remember, you only need half a mil, so 500 mils a day, to release and eliminate all of your body waste. So some of us urinate more, some less. Hopefully not much less, though. You can empty your bladder prior to micturition reflex by doing that Valsalva maneuver, and this is going to initiate it by stimulating the stretch receptors, because by pushing and compressing the pressure in the bladder, you stretch it and distend it out, which causes it to think that it's, it doesn't really recognize, oh, am I stretching because of fluid or am I stretching because of pressure? It just sends signals up to the pons. And that is urination.